So I guess this is the last session right before we move to bar, drinks, and a lot of networking. So really looking forward to that. So first, just want to say, thank you for the opportunity to be here. It's so great to be with so many people that are focused on the same thing in terms of bringing transformative cell therapies, gene therapies to patients. So I just want to quickly introduce you to Neximmune. So this is Neximmune is based off of an AIM platform technology. This is an artificial immune modulating technology that, like many in the field here, are trying to drive cell-mediated responses. So hopefully as I describe this technology and the so what about it, you'll see where we think there is unique differentiation with this platform. Obviously, our goal here is to drive cell-mediated immune responses. And really how we're doing this is sort of based off of a central platform, nanoparticle platform technology. This nanoparticle has been precisely designed to act as a synthetic dendritic cell to direct T cell function, delivering precise instructions to T cells. So this dual signaling mechanism we think is actually really important and central to the activity of the platform and the products that we're developing. So the way this works mechanistically is antigen-specific engagement. So this is HLA engagement, finding the T cells of interest. And then in the case of cancer, we want to deliver a co-stimulatory signal. So in here, we want to expand and drive proliferation of multiple antigen-specific T cells that can recognize their respective targets on cancer cells, engage and kill tumor, and obviously to clear cancer and to clear tumor cells. In the case of autoimmune disease, we do just the opposite of that. So again, we're engaging in an HLA antigen-specific way, but now we really want to target these bad-acting, antigen-specific, autoreactive T cells and drive them either into suppression or to self-destruct, so ultimately to eliminate these bad-acting, autoreactive cells. Really, at the end of this is very much about using natural signaling biology to direct T cell function. The T cells that we're expanding or activating or suppressing are not engineered, so it's very much about using the endogenous T cell repertoire. So underneath the hood of this is the construct, so this is what the nanoparticle looks like. There's a core nanoparticle. It's made of two different compositions. One is a paramagnetic nanoparticle, and the other is a biocompatible polymer. To that nanoparticle, we construct these really important dual signaling proteins. The first is a signal 1, so that's the HLA antigen presentation to find the T cells of interest. And then the second signal is the co-stimulatory signal. In this case, we're using an activation signal in our lead clinical program, but this can be swapped out to deliver different types of instructions to drive a different function to a specific therapeutic goal. To the right, you see what the nanoparticle looks like. So it actually bypasses the host dendritic cell or any type of dysfunction in the immune system and goes straight to the CD8 T cell of interest and activates through this dual signaling mechanism. It's very much about a modular approach, and we've actually designed it to be clinically applicable across multiple different therapeutic areas. So here I just described the fact that we have multiple HLAs, which is about eligibility, but we also can switch out these signal 2s to derive very specific function depending on the goal that we're trying to achieve. So ultimately, we're just building these very custom nanoparticles. What matters is now how do you take them into the clinic? So it's about the targets that we actually are going after in each indication that we pursue. So what we have recently demonstrated is the ability to rapidly develop a new product by identifying those targets and then validating those targets. We have a number of world-class collaborations in academic institutions to help us do that. We're also employing artificial technology to help us find go through large data sets to find specific targets that could be of interest in future diseases. So we marry those collaborations and the target discovery aspect with a way of exploiting this platform technology in a slightly different manner where we can actually validate that these targets will generate a CD8 T cell response. Those T cells have killing machinery and that they can actually engage, target, and kill. So through that validation process, it just takes a couple of months to do a high throughput screening. More recently, we went from the middle, which is the target identification validation, to the new product against 5 different antigen peptides for our HPV solid tumor program between November of selection and filing the IND in the second quarter of this year. So just to kind of put this all together, this nanoparticle actually plays out in its use in 2 different modalities. The first, and what's in the clinic today, is an adoptive T-cell product where we're using the nanoparticle to enrich and expand these very rare populations of antigen-specific T-cells to make our T-cell product. And then the other modality is an injectable modality where we're just simply changing out the core material. This is preclinical. We plan to file the IND in the last half of next year. So we're learning a lot from the field, I think, as everybody here is doing, right? What I've put together here is what we think are 4 different attributes that are a composite that we think are unique and differentiated about this type of technology that plays out in each of the products that we actually manufacture. So the first is the ability to simultaneously go after multiple targets. In cancer, that matters because we're addressing tumor escape pathways and mechanisms. But for autoimmune disease, it also matters because you're trying to prevent epitope spread. You need to go after the multiple bad-acting antigen-specific cells and engage them. When we want to break tolerance, and when we think about the cancer application, we want our antigen-specific T cells to have the right kind of killing machinery. So our T cell product includes phenotypes that both have effector functions, so effector memory cells, as well as the ability to establish long-term immunologic memory. That's the T stem cells and central memory cells. When we think about autoimmune disease, that's where we're going to induce tolerance. We're actually able to engage those T stem cell populations and drive them either into suppression or to ultimately to eliminate them. You can think about in autoimmune diseases, those are the cells that are almost acting like a cancer stem cell, and they're repopulating effector cells and these autoreactive T cells. So if you really want to get at the root cause, it's important to delete those or suppress those very specific targeted cells. Fundamental to the whole thing is this very much about driving the natural immune response. We think that's very important by employing this natural signaling biology, but also so that we can reduce and or eliminate the risk for these on-target, off-tissue toxicities. So now it's really translating into products and how we're moving forward. This just gives you a snapshot of the pipeline. Our lead program is in AML patients who have relapsed post-stem cell transplant. These patients have a very poor prognosis. It's a very aggressive disease. More recently, we cleared our IND to move into our first solid tumor indication. This is in HPV-related malignancies. We're looking forward to moving this into the clinic in the next couple of months. On the bottom of this slide, you'll see that injectable nanoparticle, series 100, 200, 300, will start first with oncology because they actually share the same signaling proteins. We're just swapping out the core, so we're very much looking forward to getting these into the clinic. I'll spend a couple of slides to talk about the lead programs that we have in the adoptive cell therapy. So to start with AML, these patients have a very aggressive disease and a very poor prognosis once they've relapsed post-stem cell transplant. There are no approved therapies. The current standard of care is a DLI. A DLI might give a patient somewhere between 18 to 20% response, but with that comes some very significant toxicities such as serious infections and graft versus host disease. So even if a patient responds, they may find themselves in a hospital under pretty significant medical management for these severe toxicities. So our goal here is to deliver transformative outcomes for these patients superior to a DLI, but to decouple and lower these burdensome adverse events. We're in the phase 1-2 dose escalation study, so we've got 4 doses that we are evaluating. We're currently in dose level 4, but here I just share some of the early data from the recent cohorts. What we've seen and what we expected is NEXI 001 is well tolerated. It is an endogenous T cell product. We have seen 1 grade 2 CRS, which resolved within 24 hours. We're also seeing evidence of the mechanism of action of this technology that you would expect. We're seeing multiple antigen-specific T cells proliferate and persist in the patients, not only in blood, but they're also trafficking to the bone, and we see them also maintaining that important phenotype for anti-tumor activity, but also for immunologic memory. To the right, you'll see just some early clinical activity. We're seeing signs of clinical benefit, reduced tumor burden. We're seeing bone marrow recovery. We're seeing improvements in transfusion burden, and patients are ultimately feeling better. Here you'll see dose levels 1 through a couple of patients in dose level 2. And just want to point out, we think there is potentially a signal of dose response and why we're very encouraged to be in dose level 4 right now. So here you can see to the left is the first patient we dosed, that's 50 million cells, and the second patient to the right is one of the patients in dose level 3. This patient had a response, a morphologic complete response. There was still MRD residual disease. It was 0.05%. So we're right on the cusp, we believe, to offering real benefit to these patients as we get into and complete dose, well, we are in to complete dose level 4. Turning to NEXI-003, there are a number of significant cancers that are driven by HPV, particularly these high-risk strains of E16 and E18. There is also evidence of activity, or early proof of concept, from many of you who have gone before us. There's been positive data in TILs, and we also have seen some successes with single targeted agents. This is why we believe we have the opportunity to provide a deeper, more durable response by going after 5 different targets that cover E16, 18 high-risk strains, E6 and E7 high-risk antigens, as well as survivin, which is a cancer survival protein. We look forward to getting this program into the clinic and starting to generate data soon. So with that, I'll turn back to this AIM injectable program and maybe just kind of point out a couple of interesting features that make us really enthusiastic about bringing this technology forward. If you think about what's the same in the nanoparticle that we use to manufacture our T-cell product, we're actually expanding, we're targeting and expanding multiple antigen-specific T-cells. So the injectable does the same thing. Also, the T cells that expand in our manufacturing process that include this mix of important phenotype for activity and immunologic memory, we also see expanding by using the in vivo application. So same antigen targeting, same type of phenotype of the T cells that are expanded. The difference is we're doing it in the patient. So think about what that would be like if you're driving a T cell therapy in vivo. You know, the fact that we could go both broader to patients and speed to access to patients, we think will be very important in terms of bringing new therapies. We think it could be disruptive not only as a multi-antigen specific standard of care, but also added on top of other standards of care that are not antigen specific. So whether we're going after cancer in terms of breaking tolerance or in autoimmune disease to establish tolerance, we think this kind of technology will be transformative for patients. So lastly, I'll just summarize. We have a novel approach that is deliberately designed to direct T-cell function, and we think that drives a cell-mediated response. We're seeing evidence of that in our clinical programs. It's been really fun for us to start to see the data come to life. Early data in NEXI-001 is encouraging. We believe that we will be able to deliver something truly transformative to patients with thinking about a completely different risk-benefit profile. NEXI-003 is our multi-antigen specific product. We think, again, the ability to deliver a deeper, more durable response by targeting multiple HPV-related antigens plus survivin. And again, I look forward to bringing that forward. We've also demonstrated the ability to move very quickly from that custom nanoparticle to a new product just in months in our more recent IND, which is the HPV program. And of course, the injectable, literally creating, expanding T cells in vivo is very exciting. We've got a lot of investigators that are starting to look forward to taking this technology into the clinic. Thanks.